genetic studies on the Sinhalese is part of population genetics investigating the origins of the Sinhalese populations today. Studies looking at the origin of the Sinhalese have been contradictory. Older studies suggest a predominantly Tamil origin followed by a significant Bengali contribution with a slight northwestern Indian contribution, while modern studies using more sophisticated testing point towards a predominantly Bengali contribution and a minor South Indian Tamil and north-western Indian contribution respectively. All studies agree, however, that there is a significant relationship between the Sinhalese and the South Indian Tamil, Keralite, Bengali and other ethnic groups native to the Indian subcontinent, and that there is a significant genetic relationship between Sri Lankan Tamils and Sinhalese, and being closer to each other than other South Asian populations. This is also supported by a genetic distance study, which showed low differences in genetic distance between the Sinhalese and the Tamil, Keralite and Bengali volunteers. Predominantly Bengali origin. An allopolymorphism analysis of Sinhalese from Colombo by Dr. Sarabjit Mastanan in 2007 using Tamil, Bengali, Gujarati, and Punjabi as parental populations found different proportions of genetic contribution. A genetic distance analysis by Dr. Robert Kirk also concluded that the modern Sinhalese are most closely related to the Bengalis. This is further substantiated by a VNTR study, which found 70 to 82% of Sinhalese genes to originate from Bengali admixture. D1S80 allele frequency is also similar between the Sinhalese and Bengalis, suggesting the two groups are closely related. The Sinhalese also have similar frequencies of the allele MTHFR 677T to West Bengalis. A test for Y chromosome DNA haplogroups conducted by Dr. Tumas Kivizald on Sinhalese of Sri Lanka has shown that 23% of the subjects were R1A1A positive. Also in the same test, 24.1% of the subjects were R2 positive as subclades of haplogroup P. Haplogroup R2 was also found in a considerable percentage among Bengalis of India. Sample size used was 87 subjects. These findings are compatible with the historical chronicles, the Math of Amsa and Dip of Amsa, which describe a Vanga prince from Sinhapura in Lala of being an early settler of Sri Lanka and the progenitor of the Sinhalese. The Vangas are generally identified as Bengalis. On the other hand, Lata is identified with modern-day Gujarat and Sinhapura with modern Sihor in the Kathiawar Peninsula of Gujarat. Furthermore, the Mahavamsa states that Vijay landed first at Saparika, while the Dip of Amsa mentions Sapara and a further intermediate port, Urukaka. Vijay's grandfather was reputed to be a lion, and lions have not lived in Bengal in historic times, while they have in Gujarat, so it was possible that the lion image was borrowed from there. A study in 2007 found similar frequencies of the allele HLAA*02 in Sinhalese and North Indian subjects. HLAA*02 is a rare allele which has a relatively high frequency in North Indian populations and is considered to be a novel allele among the North Indian population. This suggests possible North Indian origin of the Sinhalese. Linguistically, the Sinhalese are closer to North Indians than South Indians, as the Sinhala language is a member of the Indo-Aryan languages. On the other hand, South Indians speak languages belonging to the Dravidian languages. The Sinhalese therefore can trace a connection to the North Indian origins through this. Not only this, but the Sinhalese predominantly follow Theravada Buddhism and for centuries maintained strong connections with northeastern India, while it too was predominantly Buddhist. This further strengthens the connection of migration between the two well after the believed initial North Indian migration to Sri Lanka. Relationship to other ethnic groups in Sri Lanka. A study looking at genetic variation of the FUT2 gene in the Sinhalese and Sri Lankan Tamil population found similar genetic backgrounds for both ethnic groups, with little genetic flow from other neighboring Asian population groups. Studies have also found no significant difference with regards to blood group, blood genetic markers and single nucleotide polymorphism between the Sinhalese and other ethnic groups in Sri Lanka. Another study has also found no significant genetic variation among the major ethnic groups in Sri Lanka. This is further supported by a study which found very similar frequencies of alleles MTHFR 677T, F22021A and AMP F51691A in South Indian Tamil, Sinhalese, Sri Lankan Tamil and more populations. Relationship to East Asians and Roma. The Sinhalese are likely to have received little or no genetic flow from neighboring East or Southeast Asian populations, and have closer affinities to Western Eurasia. This is supported by a study looking at protein and blood group loci that suggests the Sinhalese are closer to Iranians and Afghans than to Mongoloids, and comparisons of root and canal morphology of Sri Lankan mandibular molars. Skin pigmentation. In 2008, a study looked at SLC24A5 polymorphism, which accounts for 25 to 40% of the skin complexion difference between Europeans and Africans and up to 30% of skin color variation in South Asians. The study found that the Rs 1,426,654 SNP of SLC24A5, which is fixed in European populations and found more commonly in light-skinned individuals than dark-skinned individuals, has a frequency of 50 to 55% in the Sinhalese and 25 to 30% in Sri Lankan Tamils. This allele could have arisen in the Sinhalese due to strong Northeast Asian genetic admixture. Further migration from North India are strong selection factors.